state that nearly 14 million years ago expansion started. Wait, the earth began to cool, the autotrophs began to drool, Neanderthals developed tools, we built a wall. We built the pyramids, math, science, history, unraveling the mystery that all started with a big bang.
What is engineering? It consists of an artificial meeting of molecules of DNA for the purpose of insulating genes or DNA fragments, cloning them and introducing them in another genome so that they express themselves. The genetic engineering can be described as the formation of new combinations of genes by the isolation of a fragment of DNA. It is called a genetic engineering to a series of skills that allow the program genes transfer between different organisms. When the new genes are introduced in the plants or animals, the resultant organisms are called transgenic. They are genetically modified organisms. The application of genetic engineering. Genetic engineering in animals. In the research laboratories, there is investigated the creation of new races of animals by means of skills of genetic manipulation. The first step have happened obtaining clonal animals trying to obtain new more resistant and profitable races. Genetic engineering in the plants. The first plant developed from the plant of the tobacco. By means of the genetic engineering, there have been obtained plants resistant to virus, bacteria or insects. Genetic engineering in the bacteria. The bacteria are the living beings most used in the genetic engineering, by the extraordinary capacity of adaptation to different environmental conditions. They are used to prepare and to treat some illness. The most used bacterium is the Escherichia coli. Genetic engineering in human beings. It is the alteration of the genotype of an individual with the intention of choosing the phenotype fruit starts of the conception. The human engineering tries to treat genetic illness and increase the resistance. The European Council prohibits the cloning with experimental reproductive ends in human beings.
The technique of recombinant DNA is to take DNA from a first subject and the DNA from a second subject and combine it to create a new living being with the DNA of both. When you do this, you have a recombinant DNA. This is possible because genetic code is universal. This means that all human beings and living things have the same genetic code. There are 3 types of enzymes and each one cuts the DNA in a different way and only with a specific code. The type of enzyme that cut the DNA is called restriction enzymes, and there is another type of enzyme that join the DNA and is called a ligase enzyme.
What is a restriction enzyme? It's an enzyme from bacteria that can recognize specific base sequences in DNA and cut the DNA at a specific site. A restriction enzyme acts as a biochemical scissor. This is also called a restriction endonuclease. Bacteria use restriction enzymes to defend against bacterial viruses called bacteriophages. When a phage infects a bacteria, it inserts its DNA into the bacteria so that it might be replicated. The restriction enzyme prevents replication on the phage DNA by cutting it into many pieces. They are indispensable tools in recombinant DNA technology and genetic engineering. There are a great variety of restriction enzymes, and each restriction enzyme recognizes a short, specific sequence of nucleotide bases and cut it. Since enzymes cut the DNA in a symmetric manner, they create a single-stranded position called sticky ends. They will be used for introducing a DNA fragment in the plasmid for other uses.
Plasmids and recombinant DNA technology. Recombinant DNA technology is the ability to develop novel genes and clone them by introducing them into new cells, using their machinery to make copies. How can we do this? In order to insert a segment of DNA into a cell without the cell destroying it, we use a vector, such as a bacterial plasmid. A plasmid is a natural occurring circular DNA that exists in addition to the main DNA. It is also double-stranded. It replicates independently of the cell's main genome. Example of plasmids. The first one is PBR322. This was one of the first plasmids used for cloning recombinant DNA molecules. It contains genes that give it resistance to antibiotics, ampicillin, and tetracycline. A DNA fragment of interest can be inserted into the plasmid by using restriction enzymes. We can use different restriction enzymes to insert the fragment at different locations. By inserting the fragment into a gene, we inactivate that gene. This is called insertional inactivation, which means that the gene will not produce the corresponding protein. Here we have the PBR322 plasmid, which includes a tetracycline-resistant gene and ampicillin-resistant gene. For example, in this plasmid we have ECORI gene, SALI gene, and PSTIC gene. By adding a DNA fragment into the PSTIC gene, we can obtain a plasmid with the PSTIC gene inactivated and the SALI gene intact. The second example is PUC18 plasmid. This is a more versatile plasmid that is commonly used as a vector. This plasmid also contains an origin of replication and an antibiotic resistance gene. But in addition to this, it also contains a B-galactosidase gene. This gene codes for an enzyme that breaks down sugars and can be used to give a distinct blue color. This gene has also been modified to have a polylinker, which can be used to fit any DNA fragment into the gene. This is the plasmid PUC18, which has a B-galactosidase gene with the polylinker on it, an ampicillin resistance gene,